Hello, hello. Is this on? あ、これ、いいですよ。はい。はい。そうですね。はい。これなんか、ポインターが出ないね。電池がない。こっち。
ポインターではなく、
進めるだけ。あ、こうなのか。
デジタルです。
あ、こうなる。なるほど。
で、多分コントロール、もし、
ええ。いいです。
レーザーポインターがコントロール L を押すと、レーザーポインターに変わります。矢印みたいな。
ああ、なるほど。これで、こっちは、操作は別にこれでいくわけ。
はい。
なんか、
ポインターモードだと、
これができないよね。
なので、そうですね、矢印モードにしておくのが安全でしょう。
どうやってするんですか？今どっち？
で得られるんですけど、矢印モードにしてあります。
で、これだとこうなるのか。あ、これでいいのか。あ、これでいいのかね。あで、これいかないや。こっちが、あ、いけるか。いけるか。いけるですか。いいですかね。あ、大丈夫。動画をクリックしなきゃいけないんで、まあいいです。
試してみます。練習してるんですよ。
これは、これでこないといけない。これいいか。まあこれは、あ、いくのか。
これでボタンが押せないわけですか？
いや、押せてる。押せてるからいいか。いいんじゃない？まあ、いいです。どうもいいのかな。入ってるかな。はい。え
Okay, after completing the master course there in 1979 and following a brief period working as a researcher at the NTT and the Ministry of Transportation of Japan, he returned to Tohoku University as an assistant professor in 1981. And then he obtained his PhD in 1986 under the supervision of Professor Yasushi Sawada with a dissertation entitled Unpredictable Motions in Fluid Systems. This work appears to be the fluid mechanics research, which I guess eventually led to his another highly influential and well-known study on the universal transition to the turbulence in channel flow as directed accretion. I don't know if he plans to talk about it, but anyway, in 2000, he joined Department of Physics of the University of Tokyo as a full professor. He worked there roughly 20 years long and retired in 2019 when he moved to Japan and served as a chair professor at the Institute for Natural Sciences of Shanghai Jiao Tong University, where he spent roughly 6 years. What he will talk today may include the works, what he did in these periods on the physics of multi-living cellular dynamics, I think. Anyway, he has since returned to Japan and now currently holds the visiting researcher positions around Tokyo. His research interests span a wide range of the fields, including statistical physics, nonlinear dynamics, soft matter, active matter, as well as biological physics, biophysics. I believe that his talk today will reflect such broad perspective and interdisciplinary approach to the problems in biological physics. Okay, thank you very much, Sano Sensei, for accepting our invitation. And everyone, please join me in welcoming him. Sano-san, the floor is yours. Thank you.
Thank you very much for very kind introduction, maybe too kind for me. Okay. I'm very happy to come back this institute, Academia Sinica, Institute of Physics. I have been visiting CK-chan several times. And also, before COVID. Okay. Yeah, after COVID, this is the first time. And today I'll talk about some biophysics related problem, but it's very much related to statistical physics and biophysics. So I hope you can enjoy, but maybe if I talk a little bit too complicated, maybe if you don't understand, you can ask a question anytime. Okay, so why we are interested in the multicellular biological system? Because this is a movie of the development of Drosophila embryo. After cell division, the cell sheet enclosing the body, and the cell sheet moves like this. Sorry. Then the shape, and later the stripe pattern appears by the gene expression. But we are interested in how this cell sheet is created, how cell sheet are moving and organized to make some specified pattern. So I show this movie because our body are composed of many bags or sacs formed by sheet of cells. This is in fact, many people say that, but I just remind that Hisao Honda, who is one of the inventor of vertex model to simulate the multicellular system, also the dynamics of soap film. And he says our body, of course, our skin are made of epithelial sheet, and our stomach and intestine, many organs are also enclosed by a cell sheet. And so the interesting question is how this cell sheet are formed starting from a few cells, and build a body through the cell division and movement and deformation. And also, cell can differentiate into different cell types. For instance, our body is made of about 200 kinds of different types of cell, like hair or skin or bone. But each cell has almost same identical genes. So, some genes are active, most of the genes are inactive. How these differentiation are controlled? And also, when our body is grown, our shape is almost similar, and just the size is increasing, and most importantly, the proportion of the cell types are maintained during the growth. So that is the homeostatic stasis. So this is a very special system and growing system. It's very different from what we know about the statistical physics. In statistical physics, we often use the simplest model, like Ising model, made of identical unit, identical spins, for instance, and uniform in 1D, 2D, and the transition often satisfies the detailed balance. But in the biological system, each element, for this case, each element is a cell, individual cell. Each cell can move by active force, by converting free energy into motion. So sometimes we call the dynamics of cell sheet as an active matter system. But in reality, our body is much more complicated because it's growing in number by cell division. Also, they rearrange the orientation and relative position and deform the shape of the cell sheet. So this gives us a lot of chance to study the interesting phenomena in biological system. あ、そっちを使いますか。はい。Okay, today's topic, mainly I talk about two topics. If time allows, I talk about third topics, but maybe I don't have enough time. So the first one topic is about the understanding collective dynamics of cell layers, cell sheet, and often our skins or some tissues making organ has some orientational order, not positional order. And there are also topological defects and cell accumulate and disperse. And there are 3D structure formations. And this, I worked in the Shanghai Jiaotong University last 5-6 years. And the second topic is about how does single cell develop into tissues. So our body in the development start from single cell, and this single cell divide and make some envelope box with some cavity inside. And this is the first occurrence of the tissue or cell sheet in growing system. So I have been working on this problem with Tetsuya Hiraiwa for several years in the collaboration with the French and German groups. So this will be second topics. If I have time, this is not understand very well, but I have been interested in how to make a useful model for cell differentiation. This is still difficult, but it's also interesting problem. Oh. Okay. The first part, I talk about the specific cell type. It's called neural progenitor cell. It's almost same as a neural stem cell. It means this cell can differentiate into 3 types of cell: neuron, astrocyte, oligodendrocyte, if there are sufficient growth factor and the environment are prepared. And in the real mass, the brain is formed after making 6 layers, and there differentiation and function, biological function appears. However, if we don't give a proper growth factor, the cell can stay in this multipotent state without differentiating. So they can divide almost infinite time. So you can store this cell in the refrigerator. So anytime you want to do experiment, you just wake up, and then you don't need to kill mouse. That was important. I worked with CK-chan on brain network on the dish, and we have to kill the mouse every week. And after killing many mice, I was very sad because I couldn't write the paper. But the CK-chan was very clever, so they wrote a lot of paper, nice paper, together with myself. I have to thank CK-chan. So anyway, so this system is much easier if you have this cell line. So they don't differentiate. So we can make the cell culture on the dish. At low density, you can see that each cell are elongated, they are touching. And on the right-hand side, you see the image of the nucleus for each cell by fluorescent protein. And each cell moves almost randomly. It moves straight, then change orientation, then change orientation. And at a higher density, cell align like this. And if you look at the motion of the each cell, so cell trajectory move along this orientational order. However, the orientation can reverse from time to time. So they exhibit back and forth motion. So this kind of orientational order we call nematic order because it's like a liquid crystal, nematic liquid crystal. There is no distinction between head and tail because cell can move back and forth, and they can align in the along the axis. So this is the nematic order. So this is a movie for the cell motion. So they cell move along the axis of the orientational order. Then you see the cell can move back and forth, and they can pass each other. So they behave like liquid, but with some orientational order. It's not solid. You know, the relative position of cell are easily slide and exchanging. And if you look at the one-dimensional motion of each cell, we can confine each cell at low density in the narrow groove created on the substrate by micropatterning. And this each trajectory is the motion of the cell along the x-axis. The vertical axis is time. So each cell, this is just overlap of many trajectory. Each cell exhibit back and forth motion. And the corrected motion looks like this. And okay, this is also movie. At high density, you see still nematic order. Cells are aligned along the local order. And here we can define the orientation of local orientation of the cell by using some image analysis technique. And since there is no difference between head and tail of the cell because they move back and forth, we can assign angle of each cell, orientation of each cell between 0 and pi. And there are singular points. At this point, we cannot define the orientation. So this is singular. And here, if we look at the orientation more closely, we see topological defect. The one type is called +1/2 topological defect. The other one is -1/2 topological defect. So if you count the rotation number, winding number around this topological defect, we can measure the angle of local cells measured from x-axis. And if you rotate on the counterclockwise direction, the orientation rotate pi. After rotating pi, it can overlap on the original pattern. So in this case, orientation angle is half of the full rotation. So we call this +1/2. On the other hand, for this kind of triangular like defect, if we measure the orientation along this rotate on the opposite direction relative to the moving counterclockwise direction. Orientation moves rotate in the clockwise direction. So the topological charge here is one half. So minus half winding number. So this red one is minus one half, plus blue one is plus one half. And you see some 3D band created on the 2D nematic order. It seems topological defect is attracting cells in this system. So it is true. If you see this kind of comet-like structure, it's plus one half topological defect. And cell density increase at the core of plus one half defect. On the other hand, cell density relatively decrease compared with the average density at the minus one half topological defect. So this is I guess this is the first time that topological defect in orientation order playing some biological role in the real experiment. At the same time, the group in Singapore mechanobiology lab collaborated with Radu in Paris. They also found a similar phenomena for the MDCK epithelial cell, where the density of the epithelial cell are controlled when cells are extruded from the plus one half defect, and the surrounding cell can invade the vacancy. So cell density control is maintained through the dynamics of topological defect in some epithelial cell sheath. So what is the effect of this topological defect? If we consider the plus one half topological defect, I said there is a comet-like structure alignment around the plus one half defect. So in conventional classification of cell sheet, people often classify cell sheet between extensile behavior and contractile behavior. It says that extensile means each cell want to extend along their long axis. In this case, each cell want to push in this direction. Then the total net motion of the plus one half defect is moving upward towards the head direction of the comet-like structure. However, if each cell want to contract along the long axis, they contract in this direction. Then this plus one half defect should move to downward. So that was a conventional classification by looking at the motion of the plus one half topological defect. And if there is no defect, there is no net force. And this kind of behavior can be easily explained by the minimum model by considering the pneumatic tensor order parameter. In 2D, theta is the angle of the local orientation, and this 2x2 tensor matrix satisfies the symmetry of pneumatic system. If you change theta to theta plus pi, this doesn't change this tensor matrix. So this satisfies the pneumatic order, and amplitude is scalar amplitude. It's 0 if it's disordered. If S is 1, it's perfectly ordered. And if there is no deformation of the pneumatic field, then Q is homogeneous. Then divergence of Q is 0. In that case, there is no active force. And if we have a deformation of a Q field, mean theta depends on spatial variable coordinate. In that case, divergence of Q is not 0. This creates active force. that is the simplest minimum model for active matter theory. And this active force created by the deformation of the nematic order create active force that can balance with the frictional force between cell and the substrate, cell and the extracellular matrix. And this was the simplest possible model. There are also many other models like active gel theory by Prost and Giovanni and Kruse. They have extra terms, but this active force is always same. Yeah. Okay. This is the some kind of a coarse-grained picture in time and space. Or it's the theta is already some average angle, and the force is also averaged in space and time. So there are some microscopic motion, but we average in time and space. Then we expect these force are slow variable. This order parameter is also changing slowly. Force distribution are also changing very slowly. Each individual motion, back and forth motion, is much faster. We average out this. And the similar situation was found in other systems like monolayer of the some bacteria, and then when the monolayer become double layer, 3 layers, and then plus one defect become a center of the multilayer formation. This is the case for the other systems, and they also observed some topological defect playing some role. And the interesting experiment came out. The Israeli group reported that hydra, mouth and foot and tentacles, the tip coincide with the position of the +1 defect, +1 topological defect. +1 topological defect is like this. So the orientation of the angle rotate 2 pi if you make one turn around the topological defect. And in the hydra, the actin filament is forming this orientational order. And the topological charge is conserved, so if you create a +1 defect, then you have to create a 2 minus 1/2 topological defect. Similar phenomena are also observed in the growth tip on the plant. In plant, each cell has the orientational order of the microtubule. So microtubule forms the +1 topological defect at the growing tip meristem of the plant. So the +1 defect are interesting. There are different kinds of +1 defects, like this radial pattern called aster, this circular target pattern is also +1, and the spiral is also +1. Just the difference is this. So we can define different type of +1 defect by changing this tilting angle, theta naught. So each angle of this element theta is measured by the angular coordinate phi plus initial tilting angle theta naught. If theta naught is 0, this is a star. If theta naught is pi over 2, 90 degrees, this gives the target button. So there are many different +1 defects. So if we induce the +1 defect in pneumatic system, what will happen in active matter? That was our question. So we can induce the Asta spiral targets by changing the tilting angle, theta naught, and then assume that if the theta, this coefficient is positive, that means the system is extensile. We can calculate the active force very easily. Then the prediction is that this scalar order parameter, S is always positive. S is increasing with the radius, so this is positive, positive. So sign is determined by theta naught. And the simple prediction is that if theta naught is smaller than 45 degrees, if tilting angle is smaller, like Asta, then force is inward toward the origin. So cells should move to the origin for smaller theta zero, mean Asta pattern or not tightly curved spiral. However, if theta naught is larger than 45 degrees, like target pattern or tightly bended spiral, and this change sign, then this become positive. So force goes outward. So cell should go outward. So this for extensile system, depending on the tilting angle, cell behavior change completely. And if theta is negative, that is for contractile case, then it's completely opposite. So this attraction or depletion at the core of +1 defect is a dichotomy, either attract or repel from the core of the defect. That was a simple prediction by the all kind of conventional theory for the active matter. So we tested this hypothesis by inducing many patterns in the same dish, for instance, by changing the tilting angle theta naught from target, spiral, and Asta clockwise, anti-clockwise. And this was designed by making some shallow reach on the PDMS. The height is only 1.2 micrometer. So cell size is about 10 micrometer. So cell can easily override. Can you see? So cell can move along this, but they can override, go beyond the reach at low density. However, when the cell become high density, they align and they move along this reach. That is to induce by applying weak external field by the 3D pattern. So this is the movie of the cell motion, showing the motion of the cell nuclei. So you see the cells are moving along the circular reach and move back and forth, but they gradually tend to accumulate at the core of the +1 defect. So this is a final result. This kind of the circular pattern cell accumulate at the core of the +1 defect. And but this is in fact contradictory to the conventional theory for active matter. So as I said before, if tilting angle is larger than 45 degrees, in this case tilting angle theta naught is 90 degrees. So in that case, cell should go outward if you believe the linear active force in active matter theory. It does not depend on the which theory you use, because all theory, conventional theory, use the linear active force term. So this is true for the, yeah? No, this is not chemotaxis. This you can test by shaking the medium or changing the medium slowly, or it does not affect ND. It's consistently attract cell. This is to test whether this observation is true or not. By changing the type of +1 defect, by changing the tilting angle from 0 to plus pi over 2 or minus pi over 2, all case the density at the core become higher. So if the simple linear active force theory is correct, and the 45-degree should be the boundary between attraction and dispersion. And this is the density distribution along as a function of radius, and the all data shows the accumulation at the core. So I can skip the detail, but so we check the local orientation of a cell relative to the induced pattern. The difference is only -4 degrees in average, -5 degrees. So they are induced by, orientation is induced by the background pattern. And there is a small chirality moving clockwise direction, some tendency. However, the averaged flow, as I said, we are interested in the slow variable, like long-term average of drift velocity. So each cells are moving back and forth, but if we make many averages over space and time, we observe the mean net flow is directing towards the core of the topological defect. And the individual cell velocity is about 0.5 micrometer per minute. And after averaging, post-graining, the net velocity is about 10 times smaller. And this result is incompatible with the simple linear active force theory. And even if we consider some kind of anisotropic friction, this cannot be described. And this shows the mean flow, net flow for different patterns, spiral, and the target pattern. So all shows the inward flow. And the radial velocity as a function of radius is inward, it's negative. So we measure the distribution of the angle at each point. And there's a peak of the distribution of the angle relative to the direction of the origin. And we measure this peak angle as a function of the tilting angle, theta naught. And this shows this red dot. And this gray region is net flow is inward. If the velocity of the angle of the velocity field is larger than plus 90 degrees or minus 90, smaller than minus 90 degrees, it means going outward. But all data shows inward flow. And if we consider some anisotropic friction, this gives these broken lines. And this black dot line is our new theory to introduce nonlinear active force that that present in our system also in the natural pattern in the cell sheet of a neuroprogenitor cell. Yeah, yeah, yeah. Ah, so okay, substrate is PDMS. The cell is attaching to the substrate, and above the cell, this is medium, just liquid. And the liquid, the viscous force between fluid and cell are much smaller compared with the traction force between the substrate. So in fact, the viscous force from the fluid is not important at all. And so if we gently mix, it doesn't affect. And what else? If we put some particle stick on the cell, then we start to see the cell particle can also aggregate to the center because it's conveyed by the transported by the cell. So okay, so maybe I should go quickly. So to explain this phenomena, we employed particle model. There is a so-called Bichek model, self-propelled particle model interacting each other and aligning each other. But here the cell align nematically, so 0 and pi are identical. That is this term. And there's a velocity reversal. This epsilon change sign with some probability. This is a velocity reversal. And cell move along in the direction of theta. And there's an effect of external field. External field, we designed the theta p pattern by design. So this we consider external field. Then we introduce soft repulsive torques between cell. If I and J particle in this kind of alignment configurations, this I cell feels the torque towards the opposite direction relative to position of J. And this can reproduce the accumulation at the target pattern. This red means accumulation, high density at the core. Blue is low density at the core. So if the velocity of the particle is not too large, and this repulsion between cell are stronger, then target can attract cell. And for Aster, it's opposite. For velocity large and smaller repulsion, and cell can attracted at the core of the +1 defect. And there are coexistence of the attractive region here. And so this can reproduce the experiment. If we only have if we don't have this repulsive force, this boundary should coincide. So in that case, two states are dichotomic. It's either attractive or repulsive. So this is a simulation of the particle model. So you start to see the accumulation of cell at the Aster pattern. And finally, density at the core become higher. And then we derive the continuum hydrodynamic model from the particle model by using Boltzmann-Ginzburg-Landau type approach. And this is useful because if we have a continuum hydrodynamic model, we can analytically predict the behavior. And most importantly, this continuum model is an equation for density, momentum, and nematic order. all are slow variables after averaging space and time. It's not measuring the spontaneous the uh, the individual fast cell motion. Which after coarse graining, this slow variable play important role. Then the system should have some symmetry. There's a reflection symmetry. In that case, velocity should change sign, but Q does not should change sign. And derivative and force should change sign. To satisfy this symmetry, it's easy to predict that we have more terms like this. These terms, nonlinear term, apart from this linear term, satisfy this symmetry. And these terms are not small because Q is scalar amplitude is almost 1. So Q, nabla Q is not small compared with this linear term. And the important thing, advantage is that we can calculate each coefficient, this coefficient from the particular model. That is the advantage of Boltzmann type approach. I skip Boltzmann equation and the Fourier mode of the density distribution as a function of radius, position and angle and time. And this Fourier mode component after truncation, we rewrite this Fourier mode in the usual form of density. W is kind of momentum, rho v. Q is a density times nematic order parameter. So it's still complicated. Sorry. But this is in fact very similar to what active matter people use for the active nematics. It's called the Beris-Edwards equation. And without this active force, this linear and nonlinear term, this is similar to the equation for liquid crystal. There is an effect of shear effect of rotation, and this coefficient can be calculated from the coefficient in particle model. So anyway, it's too complex. So just I show this some simulation case when the nonlinear active force is 0 with only linear active force term in the deep ordered state, but then it shows a nematic chaos. We can also find a homogeneous nematic state. So homogeneous nematic state, these parameters, so finally all the orientation become uniform. So topological defects can annihilate finally, but this I stop at some point. Then you see that density at the +1/2 defect is higher, density at -1/2 defect is lower when the homogeneous nematic state appears. And so we can reproduce the phase diagram by using this hydrodynamic continuum model. So +1 Asta defect, so circular defect, target pattern attracts density. And the phase diagram of the continuum model is qualitatively similar to the particle model. And what is the reason for this contradiction to the linear theory? So we introduced the two nonlinear terms, which were not considered before. And there was some paper, maybe one paper in PNAS paper by Srinivasan considered one of this term, but we have another extra term. So this one, so from this, we can predict the boundary between attraction, red region, and repulsion, blue region. And So this solid line, black solid line, is a boundary predicted by this nonlinear force theory. And this broken line is the prediction only by linear active force. So linear active force cannot explain this coexistence of accumulation both in aster pattern and target pattern in this region here. So, so they then by assuming the Q nematic order tensor is almost stationary because it's much slower in dynamics. So we can calculate the actual force, radial force, from the linear and nonlinear active force down. So the radial force can be calculated like this. Then we classify the force state, not only extensile and contractile. There is another state, this section 2 and section 4. So in our case, neuroprogenital cell belong to this. This means both circular and aster pattern attract cell. And it's not simple extensile, not simple contractile system. And if this story is true or not, we can measure the coefficient of the proposed equation by using Bayesian inference technique. We have several parameters, but we know the local Q, local mean velocity V, and we know the density. So we fitted all kinds of parameters. And this is the actual momentum net velocity. This is the predicted velocity from the model. And this is the net flow pattern predicted by this model. And this can explain the direction of the net velocity toward the radial direction, and also the amplitude of the velocity. And this technique can be also applied for the natural pattern without introducing +1 defect. We can apply the same pattern, same technique for the natural pattern just on the plain substrate, and then +1/2, -1/2 defect appears. There's no +1 defect spontaneous appear, but then we have same result, similar result. So this is a This is a conclusion of first part. So second part, maybe it can be short. Yeah, okay.
In this case, do the parameters in your model remain the same? Are they universal, or it actually varies with respect to the?
It's not universal. It depends on system. Our neuroprogenitor cell gives some number, but some cell, maybe some cell are contractile. Many types of cell are contractile. Yeah, I guess. So now we are trying to apply this method to different type of cell, like epithelial cell or endothelial cell. And we are now inducing +1 defect or ±1/2 defect to apply this kind of method.
Thanks.
Okay, if we change the interval between the ring, can we predict the velocity of the cell?
Okay, so in fact, our collaborator, Sarah Francesca, she is a Netherlands. Before she was in US, and they, in fact, they designed the +1 defect much earlier than us, and they change the spacing and height and width of the reach and tested many different parameters. And they tested 3 different type cell, but not this type cell. So they somewhat optimize the parameter. So they found if the spacing is 60 micrometer, this is good. If they're too narrow, cell can stay only one cell can stay, for instance, one between 2 ridges. That is in that case maybe external pattern is too strong and too confined. If it's too narrow, too wide, then the effect of the confinement or alignment is weaker. But the coherence level of the pattern is around 100 micrometer. So we are using 60 micrometer between spacing, and this width of a ridge is 9 micrometer. Height is 1.2. They use 1.5, but yeah, we tried to change a little bit. But if it's too shallow, no effect. If it's too high, cell cannot go beyond the ridge. The confinement is too strong. So we somewhat optimize this pattern.
For the interest time, maybe you move on.
Okay, so I already spent some. So this first part is actually the almost all the work was done by my former PhD student, Zifei Zhao and Ison Yao, and also former postdoc Li He. And the second part, maybe I can go quickly. So the second part is about the organoids. So we have question that how the cell sheet is created, starting from single cell, because our body is starting from single cell. Then by cell proliferation, number of cells increase, and the cavity, lumen, which include liquid inside. So this kind of cavity is created, and then the system deformed. To study this kind of dynamics, organoid is one of the best system because it can be made from stem cell. If you embed stem cell in a motile gel, which has a lot of different kind of growth factor, ECM, nutrient, and also ECM is soft, so they can cell can deform and make spherical structure inside the motile gel. Then if you see the brain neural stem cell, the brain organoid is formed. If you see the pancreas stem cell, it makes a pancreas organoid. Then finally, it can differentiate in and secrete insulin, for instance, and it can also make the intestine organoid. So the different kind of organoid pattern now very intensely studied in biology and biophysics. So we are interested in how these structures formed. The first part is how the lumen inside the cavity is formed. And this work was done with the collaboration with the German group in Max Planck Institute in Dresden. In making tissue, the cell-cell junction play important role because this bag of cell sheet has to keep the inside a liquid inside. Our body has to be tight for to the outside. So there is a called tight junction. Tight junction is connecting cells, and there are also already symmetry breaking between outside and inside. Inside there is a so-called apical membrane outside the basal. In apical membrane there is a specific molecule like so-called podocalcine. They are like a polymer brush with a charged particle, charged polymer. They repel each other, so cells cannot stick due to this apical membrane. And then the water can enter through the membrane if there is an osmotic pressure difference. So this process is very physical process. And the experimental group can knock out the tight junction. Here the tight junction is knocked out. The other case, wild type and some crowding knockout, still shows the formation of spherical lumen. This is the surface of the apical surface. If you knock out the tight junction, the water can much easily move across the cell layer or this gap. Then the pressure inside the lumen cannot increase. In that case, surface of apical surface looks like this dendritic star-like shape. And this phase diagram has been reproduced by the idea by Tetsuya Hiraiwa here. So we could consider just a simple free energy for the bubble, for instance, and the tension by tight junction, and the area conservation of each cell, and there may be some limit. We have to limit the minimum volume for the apical area. This pattern can be reproduced by solving some minimization of this free energy by solving this finite difference method. And also the phase field model, I did this simulation by assuming some tight junction, then the apical surface can expand at higher inner pressure with some excess area for the apical side. Then this transition from sphere to star-like shape can be reproduced. And pressure can be measured by making small hole and measuring the flow speed ejecting from the small hole. The experimenters can measure the pressure inside the lumen. Then this spherical lumen and the star-like lumen can be reproduced in this phase diagram. And we are in this work. We are also working on the phase field model, which explains the shape, arbitrary shape of each cell. If you heard about the phase field model, that's the continuous model, and the inside of cell, this U is 1, outside of cell is U is 0, and depending on this force F, this stability changes. So in this model, we can introduce cell-cell adhesion, cell division, and cell growth, and lumen growth. And we can also add spindle dynamics to determine the cell division plane. After this, we can divide the cell. And this equation is in fact minimizing free energy, but the free energy is for equilibrium concept. But this system is a growing system, non-equilibrium system. So what we are doing is introducing cell growth. We are setting target volume of each cell. If target volume is larger than the current volume, cell expand to other present energy minimum. But if the volume of each cell exceeds some critical size, we introduce the spindle dynamics and determine the division plane, and we split cell into two. Then each cell again follows the growth dynamics. Then again, after reaching some critical volume, each cell can divide. And the cell-cell interaction, we can introduce cell-cell adhesion by the interaction of the UI and UJ. So cell has a lower energy when they attract each other. Lumen formation is similar to the nucleation, classical nucleation model in the crystal growth. If the radius of the lumen is smaller than critical size, this can shrink and disappear. And if the size is larger than critical size, it can expand towards some limit, which is maybe balanced by the elasticity of ECM or cells. So these terms are added to make a free energy, and it can include the non-equilibrium growth effect and dynamics of the variable, phase variable of cells can be calculated by the minimizing by gradient descent method. This I can skip. So this is a 2D simulation and 3D simulation. So cell can divide, and blue region is ECM. Oh no, sorry, lumen. Sorry, we assume this is kind of some different liquid. So each cell are dividing. We don't see the boundary between different cells, but yellow region is the outer extracellular matrix. So in this case, single lumen is finally created, and we can test which parameter is controlling the morphology of cell. So we control the minimum cell division time, TD, and luminal pressure, pressure of the lumen inside this yellow area. Then after some time, we observe this, and at the higher pressure and slower cell growth, longer cell division time, we see monolayer. And if the cell division is fast enough, we have multi-lumen and some branched lumen. In the limit, small TD limit, we start to see the star-like shape of the lumen. And the mechanism of the making monolayer is interesting, but I need to skip this because I don't have time. So we can draw how the two parameter, lumen pressure and cell division time, is important to making final shape of the organoid. And this model has been tested to compare with the 3 different systems. This left-hand side is a model organoid made of MDCK cell line, which makes spherical lumen and monolayer. Epiblast is taken from mouse, and epiblast, the lumen formation is delayed, much delayed. And the pancreas, it depends on the outside medium. In this simplest case, it's similar to spherical organoid, but in the normal Newtonian condition, then multi-lumen and network structure appears. So So the Daniel Rivlin in Strasbourg, they changed the initial seeding number of the stem cell on the vertical direction, and looking at observe the time course of the development. So if you start from a small number of cells, it easily makes spherical lumen. If you start from many cells, like 16 cells, there are many small microlumen appears. However, this microlumen can fuse and finally makes a spherical single lumen. So this and the number of lumen increase at first, then decrease, and the same for 3 system, slightly different, depend on the some time constant. And this can be basically qualitatively reproduced by our phase field model. And this is showing the fusion process of lumen. On the right-hand side, I show the experiment for the MDCK cell. So the small lumen are now expanding, and they fuse and make 3 lumen to 2 lumen, and finally single lumen. So on the left-hand side, it looks too similar to this experiment, but I think that Fuji-san is a main worker from this simulation. So she tried many parameters or initial conditions, then find a very similar pattern. But anyway, statistically, this process is reproducible. And I don't go very detailed, but the formation of lumen is analogous to the nucleation in crystal growth. As I said, whether the initial nucleation of bubble or crystal growth is governed by the decrease of free energy, also the increase of surface energy. This is a competition between surface energy and the crystallization. Then depending on the inner pressure of the lumen, and if inner pressure is like 100 Pascal or 30 Pascal, from the initial cell division, like 2 cells, is cell divided, then the lumen can be created. So each time cell divide, then new lumen can grow, and then microlumen can fuse to make a single lumen. If inner pressure is higher, it's much easier. However, like epiblast, it is known that the leakage between lumen and outside is large. In that case, we assume the delta P inside the lumen is almost 0. In that case, the lumen cannot be nucleated until the cell number reach 8 or 10, because cell-cell adhesion is much stronger compared with the energy gain by creating lumens. So this looks very simple story, but we hope this kind of mechanism can explain the initial process of lumen growth. However, if we go further for the pancreatic organoid, this is an experiment and simulation. We can reproduce both spherical and network like multi-lumen pattern by phase field model. However, the theory or why is a branching pattern of lumen and appears in this system, we still don't understand. Also, we are still not introducing a differentiation in this organoid. So these are future remaining topics for our research. So, okay, I think I used for 1 hour. So, okay, this is the acknowledgement for second part, so I should skip third part. So this experiment was done by French group and Daniel Liebland's group, and in Germany, Angrappin bottom group for pancreas, and also Alf Nigman. Our group in Japan, Taiwan, and are composed of these researchers. Tetsuya Hirai here, also the former postdoc Kanafuji and Sakurako Tanida. So we worked on the modeling part of this. Okay, so I skip this differentiation part. So thank you for your attention.
Thank you very much for sharing the your great works. So now this place is open for questions, comments, discussions.
Thank you for the interesting talk. I have a question regarding the previous part regarding this pattern PDM substrate that caused this striking pattern formation of this accumulated density in the center. So I want to step back. If you think about if you take such a substrate and then you just put in some, say, colloidal particles here, and then the pattern is a symmetry breaking field, and basically then if you start with a uniform density and then you let the system settle, the detail balance and when you let the system equilibrate, you will have a density in homogeneity due to the symmetry breaking field. So the pattern breaks the symmetry that gives you this inhomogeneity in density. So in your case, where you have this symmetry breaking field again, and but here you don't have detail balance, but I can see where your active media, there's what breaks the detail balance is the cell development. So it's in the cell cycle. So I'm wondering, so what you are seeing is one part of the cell cycle where the cell is growing, and then you see this. But if you let it the video going, you watch a longer time, when you watch the cell contraction, or you cause the cell contraction, whether you can see this density peaks disappear.
Your question is, so if cell contract, we can explain some attraction of cell to the core? What?
Because what you're describing, this accumulation is entirely due to this cell development that gives you this non-zero force. So if you waited longer, do you restore the symmetry in time? In other words, the symmetry breaking here happens because of the driving force. And then if you wait long enough, you will restore the detail balance, right, in a much longer time.
Okay, so in fact, active force, if it exists, active force breaks the detailed balance.
Yes.
Because, yeah, the external field we applied is not directed field.
I understand.
It's
Yes.
there's no direction.
Yes.
It's just a pneumatic.
Yes.
So cell can align in both directions.
Yes.
There's no vector-like
Yes.
force. But due to the inhomogeneity of the pneumatic order tensor, inhomogeneity of alignment, that produces the active force. This active force direction can be opposite. If cells are contractile, for instance, Asta pattern should expand because outer third is more diluted. So each cell want to contract. The Asta pattern is unstable. They have to extend. So they just uniform contraction cannot explain, for instance. So there is some detailed dynamics depending on Q and number of Q and the nonlinear term. That is also I mentioned that the nonlinear term coefficient can be obtained without pattern induction. If we observe the natural pattern on the flat plane and measure the Q, net velocity, density, we infer the odd coefficient by Bayesian inference. We obtain similar coefficient for the same cell. So it means that without the external pattern, we still have this kind of effect, some nonlinear active force.
Okay, thank you.
Yeah.
So just a question regarding, so in the first part you mentioned you have also did the particle model, and then later you introduced the Boltzmann type model. So I just wonder, like, for the Boltzmann type model, you can use Bayesian inference to inference the model parameters. And I wonder whether those model parameters can be related to the fundamental, for example, I don't know, like cell-cell interaction parameters. Can those be deduced?
Yeah, okay. The coefficient of the continuum model is related to the coefficient in particle model. The qualitatively, it's consistent, but if you want to compare quantitatively, the phase diagram is slightly different. So it's still the comparison between particle model and the continuum model.
So can you actually compute the continuum model parameter from underlying particle-particle interaction model?
Yeah, we calculate this. It's not, we don't perfectly succeed, but the sign of the particle model, the magnitude of the particle model has a strong correlation with the coefficient in the continuum model. So for instance, I have something like this. Oh, sorry. For instance, this extensor parameter depends on the velocity of the particle model, depends on the repulsion strength, depends on density and the polarity of the Fourier component of noise, and coefficient of nonlinear term. One parameter, gamma 2, is always negative, and the other term depends on the velocity of particle and the interactions. There is a simplification of the calculation in the Boltzmann type calculation that makes quantitative comparison more difficult, but qualitatively, its correspondence is okay.
I have a very simple question.
Yeah.
You are using neural stem cell, right?
Yeah.
For your first part of the experiment. But we also know bacteria's colony, they also have pneumatic orders. And do you think if you grow this monolayer or bacteria on some patterned substrate.
Yeah.
Do you think we can use your phenomenological model?
Oh yeah. Okay. So
Has someone done that?
yes, myxobacteria is so people in Max Planck and United States, they observed the second layer or third layer formation is a bacterial colony of mixed bacteria. So the mixed bacteria is not swimming bacteria. They crawl, move back and forth on the substrate. Yeah. Then from the monolayer, the second layer, third layer formed, and they found the third double layer, third layer or multi-layer appears at the position of red one plus one defect. So they become plus one half defect is become a core of the cell accumulation. So this is another example for the bacteria. But for the swimming bacteria, you cannot neglect the fluid surrounding cell. Bacteria is swimming by pushing the fluid backward. So the momentum, total momentum is conserved. Then fluid has satisfied the incompressibility condition. Models usually consider only fluid density or particle density and with incompressible condition. In that case, there's no accumulation because the density is constant. But if you separate density of bacteria, density of fluid, but still maybe this swimming in suspension can be very different because conservation of the momentum. I think so. So far, I think people observe bacteria swimming inside the liquid crystal, for instance. They don't see the accumulation of cell in the topological defect in the swimming bacteria. Yeah. Because if this is conserved, so if you have inward flow, you need to go outward because the flow has to conserve. So it may be difficult to observe such phenomena. Yeah.
Okay. Sorry about this. Very nice talk. So going back to the lumen formation theory, you implement the nucleation theory. I was wondering what's the foundation for this nucleation theory, because in classical nucleation theory, you have a critical barrier to pass over, a critical size of a nuclei. But we do know even for two cells, you can form a single lumen. So how you I'm just confused how you get the critical size of a nuclei.
Okay, so biological system is in fact different from physics. So if the microlumen initiated by cell division, for instance, is smaller than critical size, this should disappear in the physics theory. But here it cannot disappear because of the autocaraxin expressed in the apical side. This is made of some polymer brush. It's repelling each other. They inside apical surface forbid cell adhesion. So even the volume of the lumen shrinks. This red region this apical surface still remains. So this prevent for instance, this can shrink to zero, but still this remained. this prevent the shrinking or disappearance of the lumen. So then when the number of cells increase beyond some number, then making an arrangement of this too many cells costs more energy. Then even for zero pressure or very small pressure, lumen can expand because it's for energetically it's more favorable. That is our explanation. So to prevent the disappearance of the small lumen, the self invented to express these optical molecules. This to stabilize maybe microlumen. So, and it is also known that tight junction can also rearrange. So even if cell layer can deform, tight junction can rearrange. It's like liquid. It's liquid phase separation. So junction is very tight, but still they can rearrange and deform, and depending on the dynamics of the lumen. That is maybe known by our collaborator by Alf Honigman's work.
Thank you for the active participation and the great discussion. Let's thank the Sano-san again. Thank you very much for the great talk. Okay, so one announcement. We will have a photo shoot after this at the lobby. So if you are interested in taking the photo with him, please stay at the lobby. And we also have the refreshment outside. Okay, thank you very much.